All right, thank you. All right, so we're going to be changing pace a little bit and away from the metabolism such as glycolysis and citric acid cycle and move more towards macromolecular metabolism. And this is an overview of chapter 17. The overview of chapter 17 through 20 is called the management of genetic information or molecular biology. And in these chapters, we're going to go over a lot of different things, such as the replication of DNA, which is we consider the continuing information because this is replicating the DNA to allow separation. During division of cells, we can replicate our DNA to allow each cell to have their own copy of the genome. And then we're going to go through transcription of RNA, which takes DNA and transcribes it into RNA. And this is considered to be the amplifying information because we're making RNA that can be used within the cell. And lastly, we're going to be amplifying information, which takes the RNA and translates it into a protein, which can be used within the cell. So today I'm going to be going over chapter 17, and we're going to be talking about DNA topology, replication, and packaging. So before we dive into replication, we need to understand that base pairs complement each other based on Watson-Crick base pairing. So as we all know, adenine always binds with thymine, and cysteine always base pairs with guanine. So we need to remember that this takes place whenever we're replicating whenever we're replicating our DNA. And another thing we need to think about is the replication of DNA conservative or semi-conservative? These are two different mechanisms. So here's a good representation of the two mechanisms. In the conservative mode, we have, in this figure, we have our dark, better to see now? I can't tell. Is it easier to see now? No? Okay. So in this figure, the dark, bold blue lines are the parental strands of DNA, or the old strands of DNA that we're going to be replicating with. And the light blue, less bold lines are the newly synthesized DNA. So in the conservative mechanism, we take our old template DNA, our parental DNA, and what we get out after replication is the same. We have our parental DNA as well as our newly synthesized copy of our DNA. However, in the semi-conservative mechanism, we take our original parental DNA and we split that so that we have two copies which are the same, in which one strand is the old parental DNA and the other strand is the newly synthesized DNA. So in order to determine which one of these mechanisms was being utilized by cells, Meselson and Stahl did experiments in which they grew cells in heavy isotope of nitrogen-15. And this labeled the parental strands of DNA on the nitrogens. And what they did, after they grew them in nitrogen-15, they switched the media to nitrogen-14 containing medium for replication. And when you do this, the newly synthesized DNA contains only nitrogen-14, and the old parental DNA only contains nitrogen-15. So what they found out was that their final replicated DNA had half nitrogen-15 and half nitrogen-14, which showed that we have a semi-conservative mode of replication. So another thing we need to look at before we go into replication is DNA topology. So most DNA in cells is supercoiled, meaning that it's twisted around itself, and I like to think of this as a telephone cord. So we have here our double-stranded helix of our DNA. So when we take off, we unhook the phone, we have our DNA helix. And then when we put the phone back on the hook, it kind of supercoils around itself. So this supercoiling results from under or overwinding of the DNA. And in doing so, it stabilizes the DNA in a more compact, condensed conformation. As seen here, this is prokaryotic DNA. It's circular, and it's kind of winding around itself, becoming more compact and supercoiled. And this DNA topology is very important for many processes: replication, transcription, and recombination. And today we're going to go over the replication portion of this. And one thing I want you guys to remember when going through this is that eukaryotes have linear DNA, and prokaryotes have circular DNA. And you'll see that this will come into play a little bit later. So there are three main ideas, variables we want to look at in DNA topology. First is twist, which is the number of helical turns in the DNA. Next is writhe, which is the number of times the helix actually crosses over itself, which is considered supercoiling. So now I want to do a little demonstration. I'll take out your rubber bands. If Tim could help me, please. Uh-oh. All right, for those of you at home that aren't at lecture, you're going to kind of miss out on this. But so take your rubber bands out and kind of hold them, stretch them out really tight, and kind of twist them between your fingers. So Tim, if you could twist it to the right. Hopefully you guys can see this. You can at least see it in front of you with your rubber bands. This is called twist. We're twisting our DNA, right? So now I'm going to go more towards Tim, and now it becomes writhe. So there's not writhe. Sorry. So now we have more writhe in our DNA, and the farther we go out, the more twist there is and the less writhe there is, and vice versa when we go towards each other. All right. Thank you. All right, so this supercoiling, as I said before, it compacts the DNA to fit inside either the nucleus in prokaryotes or the cell in, or sorry, the nucleus in eukaryotes or the cell in prokaryotes. As we'll learn a little bit later, DNA is very, very long, and the nucleus or the cell is very, very small, so we need to compact it in huge dimensions. So the one more variable that we're going to look at is linking number, and this is the total of the twist and the writhe. And linking number is always an integer, and this can be changed, as we'll see in a little bit, by topoisomerases. So as I said, before we can begin replication, the two strands of DNA must be separated. And normally, if we have our DNA helix here, when we try to unwind our helix and we separate the two strands, we're going to get a lot of twisting, and it's going to be very constricted. And DNA, it's going to be really hard to replicate our DNA this way, and it's just not good for the cells. So naturally, DNA in our cells is negatively supercoiled. What this means is that when we stretch the DNA apart, it actually unwinds, and this helps separate the strands for replication. And in doing so, this provides access to the DNA by different proteins that are involved in replication, as we will see later is very helpful. So without supercoiling, we would be forcing our DNA to be twisted up in a very unfavorable form. And that's why we have topoisomerases. So topoisomerases can change supercoiling of a DNA molecule. And here are the different reactions that topoisomerases can catalyze. The one we're going to be looking at mostly is relaxation of the supercoiling. And another one is knotting and unknotting, as well as catenation and decatenation of DNA, in which two linear circular strands of DNA can actually get linked together, kind of like you guys probably see when magicians do. You get the two rings that link together. So in each of these cases, topoisomerases cut the DNA either in one or both strands. They can rearrange the structure of the DNA and to allow for either this catenation or relaxation or actually more supercoiling to occur. And then they break, and then the seals are the broken strands are then sealed. And this is needed for both replication and transcription. And we're going to look at two different topoisomerases. There are two that exist, are topoisomerase 1, type 1, which cuts one strand of DNA. And in doing so, it changes the linking number by a factor of 1, either plus or minus 1. If it changes it by minus 1, it's considered a negative supercoil. Topoisomerase type 2 cuts both strands of DNA, and in doing so, it changes the linking number, again, by a factor of plus or minus 2. So let's look at the mechanism of general mechanism of topoisomerase 1. So what happens is first, the DNA is nicked. And when we say nick, we mean that one strand of the DNA is broken. So the topoisomerase cuts out one strand. And what happens is it moves this blue strand through the one that's cut, causing it to be relaxed, in this case to be relaxed. And then so the DNA is rotated through, and the break is sealed. So what we end up with was, in this case, is a more relaxed form of the original DNA. Now, topoisomerase I is a small monomer in humans, and it encircles the DNA. So here's the structure of human topoisomerase I, and here's the DNA molecule going through the center. So and it contains many positive charges, such that it can bind to the negative phosphate of the DNA backbone. And what's interesting about topoisomerase is actually this one enzyme does all of that entire mechanism in which the DNA strand is cut, and the other DNA strand is rotated through the break and then sealed. So it's interesting that this enzyme actually, after it makes the cut, it covalently bonds to the backbone of the DNA. And in doing so, it makes a tyrosine phosphate intermediate. What this does is it stabilizes the DNA so that this gap, it just stabilizes this nick in the DNA, and which allows for rotation of the other strand through the DNA to be sealed. Let's look at a topoisomerase I inhibitor. One example is camptothecin, which is we consider an anti-cancer drug because it actually inhibits topoisomerase I by binding to it and lengthening the lifetime of the nick DNA. Now, this is bad for replication and transcription because we don't want nick DNA to be around. It can be exposed to a lot of different agents, and it can be damaged really easily. So we don't want nicks around in our DNA. However, this is good if we wanted to use it as an anti-cancer drug because by lengthening the lifetime of the nick DNA, it stops replication, it inhibits replication and transcription and kills rapidly dividing cells. And as many of you know, cancer cells are very quickly replicating. Now let's look at topoisomerase II. What this does is it cuts both strands of the DNA. So in this figure, we have our DNA double helix. You can't see both strands, but in here there are two strands of DNA. And what topoisomerase II does is it cuts both strands of DNA here and it does so with the use of two active site tyrosines that make covalent bonds. So what happens is it cuts this DNA, and it brings the other double-stranded DNA through the break. And this requires the use of a cofactor ATP. And again, it is resealed, changing the linking number by a factor of 2. So in bacteria, gyrase is a type 2 topoisomerase, which adds negative supercoils to DNA. So it actually decreases linking number by 2. Ciprofloxacin, am I saying that right? Okay. Ciprofloxacin is an inhibitor of topoisomerase II in bacteria. It inhibits gyrase. And since gyrase is a bacterial type 2 isomerase, it doesn't affect any type 2 eukaryotic topoisomerases. So what ciprofloxacin does is that it enhances the cleavage of the DNA, so it enhances the DNA nicks, and it reduces the sealing of the DNA. And again, like I said before, this inhibits replication and transcription and kills rapidly dividing cells. So replication is actually done in many factories, many discrete sites within the nucleus. So here we have a figure of a eukaryotic nucleus. And each of these bright spots represents so proteins that are involved in replication were stained and are visualized now with these bright spots. So each one of these is considered a replication factory. And in these factories, protein machinery that replicate our DNA are stationary. And foci just means that they are localized to these specific spots. So in bacteria, there are between usually 1 or 2 different factories, different foci. However, in eukaryotes, as you can see in this figure, there are between 100 and 150 different factories. And each one of these has hundreds of replication forks, meaning that in each one of these dots, each one of these factory spots, there are hundreds of areas in which the DNA is being replicated. So what happens is, as I said, that the protein machinery for replication are stationary, and the DNA is actually reeled through these protein complexes. And this is very efficient when you have a lot of DNA around, and you'll understand this in just a few seconds. So let's look at the E. coli genome. There are 4.7 million base pairs in E. coli. And DNA polymerase, which is the enzyme that replicates DNA, does so at a rate of about 1,000 bases per second, which is very fast. And since there are between 1 and 2 replication factories in E. coli, this takes about 20 to 40 minutes to replicate the entire genome, which is very quick. However, in the human genome, we have 150 million base pairs. And the DNA polymerase actually is a lot slower than E. coli, and it replicates DNA at about 50 bases per second. So this would take, if we had one DNA polymerase working at a time, it'd take about 1 month to replicate our entire genome. However, this is not the case because we have multiple factories and multiple different areas of replication that are occurring at each factory, so it only takes about an hour to replicate our entire genome. All right, so I keep saying the word replication fork, and it's probably about time to define this. So as I said, there are hundreds of replication forks at each factory. And at each replication fork, we have our two strands of parental DNA. We have our parental DNA being separated at the replication fork. And we also have our new DNA being synthesized. And this is synthesized, again, by DNA polymerases. There are 3 steps to DNA replication: initiation, extension, and elongation. Today I'm going to be talking about initiation and extension steps. During initiation, the replication fork is formed. And replication begins at what we call origin of replication. So an origin of replication is just the site at which DNA, the synthesis of DNA is initiated. We're also going to be talking about extension, in which complementary DNA is synthesized, and it's done so in both leading and lagging strand synthesis, which we'll discuss more in detail later. So as I said, the DNA polymerase is the enzyme that replicates our DNA. However, it needs a lot of help, a lot of different factors in order to accomplish this. So our game is DNA replication. And we have a lot of different proteins that help out DNA polymerase. These are all the players that are involved. We have helicases, which break apart these 2 strands of DNA. We have single-stranded binding proteins that bind to the single-stranded DNA. We have our DNA polymerase, which synthesizes the DNA. We have our sliding clamp, which helps the processivity of the DNA polymerase. And don't worry, I'm going to be going over these in more detail in a little bit, so you don't have to worry about writing too quickly. We also have our primase, which does the initiation step of DNA by laying down an RNA primer. I'm going to be talking about telomerase in probably the next lecture on Wednesday. And then we have our ligase, which does the final step and closes up DNA replication. So let's talk about helicases. What helicases do is they separate the 2 DNA strands, and this allows access of the replication machinery to the base pairs. Here's a picture of the structure of a hexameric helicase. And all helicases are energy dependent, and so they need ATP to work. And what happens is they actually spin very quickly. And it's interesting, they're very similar. They spin similar to the F1 subunit of ATPase, which you all should be experts on by now. And they do so, they separate DNA very, very quickly. And they do it at speeds of up to 750 nucleotides per second. Now, a friend of mine told me a pretty corny pickup joke, and she said I should say it to you guys. So I don't know if you want to hear it, but I'm probably going to say it anyway. So if you're, let's say you're at a bar, or you're in class, and someone sitting next to you is pretty cute, you just go up to him and say, if I were an enzyme, I'd be a helicase so I can unzip your jeans. All right, thanks. I was really hoping there weren't going to be crickets on that one. That was too good for that, though. All right, so on to single-stranded binding protein. So after helicase separates the DNA strands, we have single-stranded DNA around. And this is not good, because what can happen is that the single-stranded DNA can actually come together and reanneal with itself. That's because that's its most favorable form. And it can also form secondary structure amongst itself, kind of like RNA does in forming hairpins. And in doing so, secondary structure or reannealing of DNA strands inhibits replication. Thus, we have our single-stranded binding proteins, What these do is they bind to the DNA, and they coat the DNA to protect it from any of the secondary structure from occurring. And this is due to the lysine that's in single-stranded binding proteins that interacts with the negative phosphate group on DNA backbone. Now, lysine is positively charged physiological pH, so it can bind to the negative phosphate. And this is not a very specific interaction, which is good because we want this to bind to all forms of single-stranded DNA. So in eukaryotes, the single-stranded binding protein is often called replication protein A, or RPA for short. And single-stranded binding proteins form many different functions in DNA metabolism: replication, repair, and recombination. We're going to be going over replication today, and later on we'll be talking about repair in further chapters. All right, on to DNA polymerases. This is my favorite enzyme in DNA replication, because I do research on DNA polymerases. And I think this is probably the most important enzyme in DNA replication. So what this does is actually synthesizes DNA by incorporating incoming nucleotides. So here we have our old parental DNA strand, and this is always called the template strand. So we have our DNA template strand here, and what DNA polymerase does is it sees that we have a thymine that needs a nucleotide to be incorporated opposite it. So here comes adenosine triphosphate. So this base pairs with thymine because of in the active site of the DNA polymerase. So the new DNTP is always added to the primer strand, which is the newly synthesized DNA strand. So this is always going to be the template strand, and the one that's being synthesized is always the primer strand. So DNA polymerases always synthesize DNA in the 5' to 3' direction. They can never go the opposite way. And this is important for a lot of different things, as we'll see later on. So what they do is the 3' hydroxide group of the existing primer nucleotide goes in and attacks the 5' alpha phosphate group of the incoming nucleotide. And in doing so, it releases pyrophosphate. And that forms a phosphodiester bond between the two nucleotides. Now, DNA polymerases are very processive. And by this, we mean that several nucleotides can be incorporated while the DNA polymerase is bound to the DNA. And this is roughly about 5,000 nucleotides per cycle. And it would be very unproductive for a DNA polymerase to come on, bind to the DNA, incorporate a nucleotide or two, come off, bind again, incorporate a few nucleotides. So we need our DNA to be very processive. And another factor that helps the processivity of our DNA replication are called sliding clamps. What they do is they clamp around the DNA, and they allow the DNA to go through the center of the protein. And they also bind the DNA polymerase, which holds the DNA polymerase in place. So this enhances the processivity of the DNA polymerase again. Over here we have figures of both the prokaryotic and eukaryotic sliding clamps. Up here is the beta dimer. I'm sure you can see why it's called that, because there are two subunits that are exactly the same. And here is the eukaryotic sliding clamp, and this is called proliferating cell nuclear antigen, also known as pCNA for short. And this is a homotrimer, so it's composed of 3 different subunits, but they're all the same. All right, on to DNA polymerases again. The structure of DNA polymerases have a common theme, and we call this a right-handed structure because it's kind of hard to see in this figure, but it does look like a right hand. So we have our palm, which is our active site, and then we have our thumb over here and our fingers as well. And what those do is they grab onto the incoming nucleotide and hold it in place. And the palm actually contains a magnesium ion, which catalyzes the reaction of the incoming nucleotide to be incorporated into the DNA. So there are actually a lot of types of different DNA polymerases. In E. coli, there are more than 5, and there are as many as 15 in eukaryotes. And they belong to about 6 different families of DNA polymerases. And you may ask, why are there so many polymerases when all they do, they just replicate our DNA. That's it. Why do we need so many of them? But they perform a lot of different functions. The normal, what we call replicative DNA polymerases, perform the leading and lagging strand synthesis. This is just normal DNA replication. Remember, there's also DNA in the mitochondria. And so we need different specialized polymerases to actually replicate this DNA. There are also some that are involved in sister chromatid cohesion, as well as damaged DNA. And here's an example, DNA polymerase eta, which is involved in damaged DNA. And I think it's wrong on your handouts. It's actually, I think it says DNA repair. It should say translesion synthesis. Sorry about that. I don't know why I put that there, but I changed it. So anyway, translesion synthesis is just a fancy way of saying that this polymerase replicates over damaged DNA. And there are also a lot of other functions that may be occurring with different polymerases. There are so many polymerases, we don't know what they all do. And there are probably many other things that are happening that we need to elucidate further. So as I said before, there are polymerases that can replicate damaged DNA. So mistakes in DNA can be very disastrous, and we're going to talk more about that in Chapter 18. But every single day, our cells are bombarded with different DNA damage. They are bombarded with chemical agents, physical agents. Spontaneously, they can be damaged, and the replication of this damage can actually result in mutations. And the mutations in between 4 to 7 key genes in our cells can result in tumor genesis and ultimately cancer. So as you can see, this is a very important process that we need to understand fully. So most DNA polymerases have high fidelity, meaning that they very rarely make mistakes. So that whenever there's a thymine in the active site in the template, we need an adenine to bind to that. And whenever, cysteine always needs to bond to, cytosine always needs to bond to guanine. So in order to prevent the mispairing of DNA, there are 2 different mechanisms that most DNA polymerases use. First, they have an active site for DNA synthesis that is very tight. And this binds both the old DNA as well as the incoming nucleotide. And this active site is very specific, and it's very tight. And when we have our AT base pair and our AT base pair and our CG base pair actually have very similar geometry. And so the active site of the DNA polymerase can only hold this specific geometry. So if we have a T in the active site and a C comes in, it's going to kick that out because it's not going to fit. And however, sometimes they do make mistakes, so they have an exonuclease site for proofreading. So like I said, when DNA is incorporated inaccurately, an exonuclease, this mispair is sent to the exonuclease site. What happens is the exonuclease comes in, and it cleaves off this inaccurate nucleotide in the 3' to 5' way, so it goes backwards compared to the polymerase action. Then the polymerase comes in and it fills in that nucleotide as well as furthers the DNA synthesis. So this exonuclease site, as well as the specific active site, enhance fidelity of polymerases even further, so that on average, up to only 1 mistake in 1 million nucleotides incorporated occur. Now you may think, oh, that's amazing. That is really amazing. However, when you think about it, we still have 150 million base pairs in our genome. So there are still a lot of mistakes that occur. And we'll be talking further in further chapters about DNA repair and again, cancer. So there are a few problems that we need to take into account when we're doing DNA synthesis. The first is that DNA polymerases cannot start synthesis on their own. They need a pre-existing primer. In other words, they need that 3' hydroxide group that I showed you before. They need that 3' hydroxide group to attack the 5' phosphate of the incoming nucleotide. So what do we do about this? There exists a protein an enzyme called primase. What that does is it lays down an RNA primer on the existing DNA template. And what this does is it actually forms a DNA-RNA hybrid, which allows for the DNA polymerase to use this 3' OH group to synthesize DNA. And most primers are about 60 nucleotides long. And you may think, well, this is not good. Why? We don't want RNA in our double-stranded DNA. This isn't good. But don't worry, later we'll explain how the DNA is removed, and again, it's going to be replaced with DNA. Another problem that exists is that, like I said before, DNA polymerase can only synthesize in the 5' to 3' direction. So why is this a problem? Well, our DNA, as most of you know, is anti-parallel. One strand goes from the 5' to the 3', and the other one, complementary to it, starts in the 3' goes to the 5' end. So in order to synthesize DNA, one strand needs, in order to synthesize DNA in the 5' to 3' direction, we need to synthesize one going one way and the other going the other way. So the strands need to be synthesized in opposite directions. But we said that our DNA replication machinery is stationary in the complex and that the DNA is moved through it. So how does this occur? This doesn't make sense. Well, what happens is that this actually results in both leading and lagging strand synthesis. So what we have at each replication fork are 2 different DNA polymerases. And we have both our leading strand synthesis and our lagging strand synthesis. So here we have our DNA, and here's our replication fork. And we have a primer laid down here, which is our leading strand, which is done in a continuous manner by our leading strand DNA polymerase. And in eukaryotes, most eukaryotes, this DNA polymerase is called polymerase epsilon. And in the lagging strand, we are again synthesizing in the 5' to 3' direction, going in the opposite way. And this is done usually by polymerase delta. But we have a problem because, in the left-hand side, we have our leading strand, which is fine. We lay down our primer, and we can synthesize in the 5' to 3' direction, following our replication fork. However, on the other side, we're going opposite direction of the replication fork. So what happens is that we actually get a looping of our lagging strand. So once this DNA polymerase gets to the end of DNA, a new primer is put on by primase. And what happens is this DNA polymerase then kind of jumps to that primer and starts another round of synthesis. And thus, the lagging strand is considered to be discontinuous. So what we have, again, is the looping is going to occur until it reaches the beginning of the next primer. And again, a new primer will be put on, and the DNA lagging strand polymerase will jump to the next primer. Now, this results in very short sequences of DNA. And these are called Okazaki fragments, after the man who identified lagging strand synthesis. So it's actually kind of interesting. Recent evidence has suggested that the leading strand polymerase incorporates nucleotides half as quickly as the lagging strand DNA polymerase. And it's interesting to speculate that this occurs because there's so much that needs to go on in the lagging strand. And the DNA polymerase needs to make up for this extra time lost. And so it does so twice as fast as the normal polymerase, so that both strands are replicated at the same time. All right, so we have our leading strand, which is being synthesized in one long strand, and this is great. However, now we have our lagging strand, which has short sequences, it has RNA primers, it has these nicks in the DNA. What are we going to do about this? So what happens is RNase H comes along. And what this does is, so we have our first Okazaki fragment left here, and we have our second Okazaki fragment on the right here, which our DNA polymerase is synthesizing. So RNase H comes in, and it also has a 5' to 3' exonuclease activity, just like our DNA polymerase. And what it does is it actually kind of chews up the RNA and replaces it with DNA. And this replacement of RNA with DNA is called Nick translation. And then our DNA polymerase can come in and fill in the rest of the DNA until it gets up to the RNase H. And then, so we have a nick here, which we need to form a phosphodiester bond between the two nucleotides. And this is done by DNA ligase. And this requires energy through either a cofactor of ATP or NAD. But we have a problem. Sometimes it's possible that the DNA polymerase can catch up to the RNase H, or it can get there before the RNase H has time to do its full job. So if this happens, so if DNA polymerase comes in and catches up to the RNA before it can be removed, is it kind of displaces this RNA and synthesizes DNA underneath this RNA. And then what's called flap endonuclease comes in, and it cleaves off the RNA. And then again, the DNA ligase is used to come in and seal this SNP. So overall, I'd like to do a little overview of DNA replication to kind of get you all on the same page. I know it's a lot of information, a lot of different proteins. I'm hoping to try to bring it all together in one figure here. So we have our double-stranded DNA, and helicase comes in and plows through the double strand, breaking it up into single-stranded DNA. And here's single-stranded binding protein binding to both sides of the single-stranded DNA. We have our leading strand here, and they don't have, there's not a RNase primer on here, but there was. And it's our normal polymerase epsilon is coming in here to replicate the leading strand. And then we have our lagging strand, which has RNA primers on it, put on by RNA primase. And then our DNA polymerase that is used on the lagging strand comes in to fill in this, to go to the next primer. And then DNA ligase comes in after the RNA primer was removed to seal the ligase, or to seal the nick between the Okazaki fragments. So this is done both in eukaryotic and prokaryotic cells. And this replication fork shows a linear DNA. However, you got to remember that in prokaryotic DNA, they have circular, prokaryotic DNA is circular. So it's actually more of a bubble, so that we have two replication forks occurring at the exact same time. All right, I went through that kind of quickly, but I'd like to show you. I think it's kind of hard to envision all this without seeing a video of it. Maybe. So I don't know if we're going to have our sound here. Probably not. So this is actually a computer simulation of what's going on at the replication fork. And on this blue protein on the left here is the helicase, which is zipping through the DNA and separating it. It does so very quickly. It's actually rotated, which you can't see the rotation. Behind, let me go here. To the right, we have our leading strand synthesis, which and the DNA polymerase. So then here we have our looping and our putting on a primer for the lagging strand, and then the polymerase comes over, kind of grabs it, and finishes the DNA in the Okazaki fragments. So it's very, very quick, but I think it's to really see how this we actually think this works. All right, well, I went through that very, very fast. So that's all I have for today.